Hello everyone. In today's class we will talk about few of the challenges in tissue engineering and few of the developments done to tackle these challenges. The topics which will be covered are scaffold fabrication, the challenges associated with scaffold fabrication in we are discussing about three different tissues, that is bone tissue, skin tissue and hard tissue. In the as a second challenge we will talk about vascularization in tissue engineering, and also we will talk about selection of proper cells. Scaffold fabrication. As we know, scaffolds are temporary support material which will enable cells to attach, grow and form a 3D structure. Hence, a scaffold must meet the chemical and physical properties of the natural ECM, and also scaffold must degrade over a period of time so that the tissue regeneration to take place. As we know, different tissues have different regeneration time. For example, skin tissue regenerates faster than the bone tissue. Hence, scaffold must meet the regeneration time of the tissue of our interest and biocompatibility of the scaffolds. It is must that scaffold should not cause any toxicity to the host cells. In the 80s, when pioneers in the field, when they proposed polyhydrides for biomedical application, the scientific community thought that these synthetic materials would cause toxicity to the host cells. This belief was persistent until 1996, until FDA approves first synthetic polymer for biomedical application. Hence, it is necessary to see the compatibility of these scaffolds to the host cells. Let us begin with the bone tissue. As we know, bone is a hard tissue, hence a scaffold which we fabricate for bone tissue engineering should have high mechanical strength, and it is explained in three different steps, which is known as biomechanical paradigm of bone tissue engineering. The mechanical strength of scaffold should match with the our natural bone tissue. At the same time, it should not induce stress shielding effect, which is prevalent in metallic implant. What happens in metallic implants is, due to their high mechanical strength, all the load will be taken up by the metallic implants. As you see here, this is the metallic implant, and this metallic implant, due to their high mechanical strength, they take up all the load, and the region which is near to the metallic implant fail to take the load which is necessary for it to take. So, as you see, the region further from the metallic implant has higher bone mass compared to the region which is near to the metallic implant. This region does not have sufficient bone mass which is supposed to possess. Hence, eventually the regenerated bone will fail to take up the load. Hence, it is necessary that the scaffold which we prepare should not induce this stress shielding effect. Second step is, the mechanical property of the scaffold should be in such a way that it induces the scaffold to bone mechanotransition. Mechanical stimulus provided by the scaffold induces the tissue differentiation. In the undifferentiated stem cells, the mechanical stimulus causes the differentiation of the cells, whereas in differentiated cells it leads to the matrix production by the differentiated cells. And in the third steps, as the host bone tissue regenerates, it takes up the load as the scaffold degrades. Recent development is degradable metallic implant. In this study, what they have done is, they have used magnesium alloy and to see their degradation profile, and this image is the degradable polymer, and this one is the magnesium alloy. In vivo staining using calcine green. As you see here, the region which is near to the magnesium alloy has sufficient bone growth formation compared to the degradable polymer, and here are the compressive strength and elastic modulus of cancellous bone and cortical bone are given. And second second most important parameter while designing the scaffold for bone tissue engineering is the porosity. Bone is highly porous in nature and it is defined in 3 different terminologies: pore size, pore volume and interconnectivity between the pores. While designing the scaffolds for bone tissue, we should consider all these 3 parameters, and as we know the bone is having high mechanical strength and it at the same time it is highly porous. So, balancing these two is a real challenge in bone tissue engineering. Let us go to the skin tissue. This is the general structure of skin tissue, the upper epidermal layer followed by the dermal layer. If the skin injury is limited to the epidermal layer, what happens is the fibroblasts present in the dermal layer, they migrate towards the epidermal layer and they start differentiating and secreting extracellular matrix, and thereby they fill up the gap. If the skin injury is very deep wherein the dermal layer is also lost, then the clinical interventions are necessary. Here are a few challenges listed for skin tissue engineering. Scarring of the tissue. As I explained in the previous slide, the fibroblasts present in the dermal layer, they migrate towards the skin injured site and they start secreting the extracellular matrix, and what happens is they secrete collagen in excess than what it is required. This excessive collagen, it cross links and contracts, and that leads to the scarring of the tissue, and avoiding such a scarring of the tissue is one of the challenges in skin tissue engineering. As we know, skin is the outermost layer of our body, preventing from microbial infection. Hence, when we prepare scaffold for bone skin tissue engineering, it is necessary that it should have antimicrobial property. And also the scaffold which we prepare for skin tissue engineering should be a hydrogel, should able to keep the environment moist and at the same time should able to absorb the exudates from the wound. Let us go back to the previous slide. As you see here, the epidermal layer and the dermal layer, they are connected with the basement membrane. This basement membrane is rich with extracellular matrix secreted by the fibroblast cells, and this basement membrane is involved in several functions of the skin tissue, and reconstruction of this basement membrane is one of the challenges in skin tissue engineering. We will talk in great detail about the vascularization and other complicated challenges such as reconstruction of the skin appendages, thermoregulation, touch and excretions are few of the challenges in skin tissue engineering. In the scaffold, they try to see whether they can able to produce this sweat gland. What they have done was they have isolated epidermal progenitor cells from the dorsal region of the mice, and they have mixed with the plantar region dermal ECM, which has cues for the sweat gland formation. As a control, they have used dorsal region ECM, which has cues for the stratified epithelium formation, but not for the sweat gland. And this mixture they fed to the 3D bioprinter, and they try to see whether they could able to produce this sweat gland or not. And the third tissue which we will be talking about is the heart tissue. These are the few challenges listed in heart tissue engineering. We will talk about only those which are related to the scaffold fabrication. Electromechanical integration between the transplanted engineering myocardial tissue and the host myocardium is one of the challenge in heart tissue engineering. Recent development is the usage of conducting polymers. For example, polyaniline is a conducting polymer, and the conductivity of this polymer is dependent on the proton based doping process, wherein what will happen is the imine and amino groups of aniline are protonated in the presence of protonic acid. And there are few challenges associated with this conducting polymer. When we implant this conducting polymer in vivo, the dopants which keep the imine and amino groups protonated, they are lost in the process called de-doping. That leads to the decrease in conductivity of these conducting polymers, and it will not last for longer time. So, another challenge associated with polyaniline is the amine groups of aniline are lost at physiological conditions. So, in this study what they have done is they have used chitosan. Chitosan is a rich source of amine group, and they fabricated chitosan film, on top of which they polymerized aniline in the presence of phytic acid. And as we know, the chitosan plays a role of amine group source, and they try to solve the problem of deprotonation. And another problem is contraction ability of the scaffolds. As we know, the heart is highly contractile in nature. Producing the scaffold which has the contracting ability is one of the challenge in heart tissue engineering. And the another challenge is full coaptation. As we know, heart is made up of valves. These valves make sure that the blood flows in a right direction, and these valves are able to contract and relax. Producing such a valve which can able to contract and relax is one of the challenge in heart tissue engineering. As we all know, vascular network mediate gas exchange, they excrete the metabolic waste and they supply the nutrient. What will happen if we prepare a scaffold without vascular network within it? The diffusion process will take place. But however, the diffusion is limited to 100 to 200 micrometer of distance. Beyond that, the cells starve of nutrients and oxygen, and they eventually die. Hence, it is necessary to include the vascular network when we design the scaffold. The ideal engineered vessels must be able to withstand physiological pressure without any leakage, and they should not be thrombogenic, they should not elicit any immunological response, and they must be economically viable. And there are several strategies to introduce vascular network into the scaffold, and can be broadly classified under 4 categories, that is scaffold design, supplement of angiogenic factors, in vivo prevascularization and in vitro prevascularization. Let us discuss each in detail. The first one is scaffold design. One of the major prerequisite for introducing vascular network into the scaffold is porosity of the scaffold. The scaffold must be highly porous and highly interconnected. And there are several strategies through which we can prepare the porous scaffold, and few of the techniques are listed over here. For example, gas foaming, electrospinning, particulate leaching, freeze drying, phase separation and microfabrication. The second approach is supplement of angiogenic factor. Just by supplying the growth factor which are involved in angiogenesis, we can achieve the vascularization in tissue engineering scaffold. Few of the growth factor which are involved in angiogenesis are listed over here. There are 2 ways through which we can supply these growth factors, one by release by diffusion, which is a direct method, and the second one is release by cell demand. This is an indirect method. Let us talk about these methods in brief. Release by diffusion, in this method we supply the growth factor which are directly involved in angiogenesis, for example VEGF, but we do not know how much exact amount of these growth factor need to be supplied for vessel formation. Hence, what happens is that leads to the unhealthy vessel formation, as you see in the upper image. The second method is release by cell demand. In this method you supply the growth factors to the cells, and these growth factor in turn stimulate the cells to secrete the angiogenic growth factor. Thereby you can control the release of these angiogenic growth factor, thereby we can achieve the healthy vascular network, as you see in the lower image. The third approach is prevascularization technique. There are 2 approaches, one is in vitro approaches and the in vivo approaches. Let us discuss each in detail. First one is the in vitro approaches. There are 3 approaches: cell seeding, generation of spheroids and cell sheet technology. The first one is cell seeding. In this approach you seed the endothelial cells into the porous scaffold and incubate it over a period of time. Eventually what will happen is there will be vascular network formation within this porous scaffold, and then you implant at the site of interest. The second approach is a generation of spheroids. Spheroids are structurally 3-dimensional arrangement of cells with intensive cell to cell and cell to matrix interaction. There are several ways through which we can achieve the formation of these spheroids. if we choose endothelial cells and tissue specific cells for the formation of spheroids, what will happen is over a period of time there will be a vascular network formation within these spheroids. Once the vascular network forms within these spheroids, then we can transfer these spheroids into the scaffold, and then thereby we can achieve the vascular network into the scaffold. The third approach is cell sheet technology. This technique is devoid of any scaffolds. In these techniques what we use is thermosensitive polymer, for example polyisopropyl acrylamide. At 37 degrees Celsius, these thermosensitive polymer they allow the cell adhesion. So, at 37 degrees Celsius you seed endothelial cells and tissue specific cell onto these thermosensitive polymer and incubate it over a period of time until it reaches the monolayer formation. Once the monolayer is formed, you decrease the temperature to 20 degrees Celsius. What happens is at 20 degrees Celsius these polymers they swell and the cell adhesion property of this polymer is lost. That leads to the peeling off of these monolayer from the polymer surface, and these monolayer are stocked and then transferred to the site of interest. Thereby we can achieve the vascular network formation at the site of interest. The next approach is in vivo approaches. In this there is a technique called arteriovenous loop formation. As you see here, the synthetic vessel is connected to the artery and venous surgically, and this after surgically connected to the artery and venous, this scaffold, this setup is implanted in vivo. As you see here, synthetic vessel is looped inside the scaffold. Once we implant inside, over a period of time there will be a sprouting of capillaries into the scaffold, as you see over here. Once there is a sprouting of capillaries into the scaffold, you surgically remove from that site and implant it to the site of your interest, that is injured site whatever. Thereby we can achieve the vascularization into the scaffold. Which is an efficient strategy. As we know, all these techniques come with certain advantages and disadvantages. For example, in case of scaffold design, it is easy to fabricate, but again the disadvantages, it relies on the ingrowth by host vasculature. In the second method, in vitro prevascularization, it does not rely on the in vitro ingrowth by host vasculature, but again the perfusion rate is low compared to the in vivo prevascularization. But in case of in vivo prevascularization, though it does not rely on ingrowth by host vasculature and it involves very rapid perfusion once after implantation, but again this method involves the surgery. The fourth method is angiogenic factor delivery. This approach has given a promising result, but again it depends on ingrowth by host vasculature. So, it is difficult to say which strategy is an efficient one. Again there are few other challenges in vascularization. For example, it is difficult to construct a tissue engineering scaffold with smaller capillaries whose diameter is less than 1 mm, and perfusion and vascularization is a significant problem in metabolically highly active organs like heart and liver. For example, in heart the intercapillary distance is around 20 meter. Achieving such a highly vascularized tissue is a challenge. We next move on to the third challenge, that is selecting proper cell source. In this section we will talk about different types of stem cells which we have exploited in tissue engineering applications. As we know, stem cells have the potency to differentiate into many number of cells, and we will talk about few of these stem cells which have been exploited in tissue engineering. The first one is mesenchymal stem cells. Mesenchymal stem cells are multipotent adult stem cells. These stem cells are able to differentiate into several cell types of the body, but not all. These mesenchymal stem cells can be isolated from different parts of the body, for example bone marrow, adipose tissue and dental tissues, but the problem with mesenchymal stem cells, they are multipotent but not pluripotent. The second stem cells which we are talking about is human embryonic stem cells. These embryonic stem cells are isolated from inner mass of the blastocyst, and these stem cells are pluripotent in nature. That means they are able to give rise to any cell types of the body, but the problem associated with embryonic stem cells is the ethical concerns, as we are extracting these stem cells from the embryo. And the recent development is induced pluripotent stem cells. What they have done is they induce pluripotency in the normal somatic cells. What they have done is they have introduced 4 genes into the mouse fibroblast cells, and thereby they could able to reprogram these somatic cells into the stem cells. But the problem with stem cells is their proliferation capacity is very low in 2-dimensional cell culture plate. So, it is difficult to harvest required number of cells that is required for our tissue engineering application. There are again certain advancements in conventional cell culturing techniques. For example, exposing stem cells to the hypoxic condition led to the improvement in their survival. So, such advancements have been done quite recently. The another problem is maturity of these cells. These stem cells they have largely differentiate into immature cells with variability in structure and function. And another problem which we face in cells is producing clinically relevant number of cells. For example, human myocardium consists of 10 to the power of 9 cells. Generating such a huge number of cells is again a challenge in tissue engineering. Thank you.